Welcome everybody. If we come to some semblance of order, it is gratifying to see that the free food has made no difference whatsoever to the attendance this week, and that you will all be here next week for the next Grand Round. It warms the cockles of my heart. My name is Tom Fowler and I'm the chair of Grand Rounds. Thank you very much for coming. Once a year we join forces with DCAT to host their annual lecture. I'm not going to steal Ewan's thunder. He's going to introduce the speaker in a short while. But I've been asked if we can hand over the first couple of minutes to a couple of the medical students who wish to talk to you about the Medic Insight program, which is an incredibly important program of providing experience to our school colleagues, children, adults, to give them experience of what it's like in that hospital. So they're going to talk to you about that because they really need your help. On you go.
Hi there, thank you Dr. Farden and Professor Pearson for letting us come and speak for a wee minute. Basically, as Dr. Farden just said, we're a student-run organisation that aims to provide work experience for high school pupils. So they come along to us in June and they shadow various clinicians and surgeons and things in different departments. They also get tutorials from us, and it's a really useful resource that the university has. I took part in it when I was at school and I really appreciated it. I think it was a big thing that swayed my decision to come to Dundee.
So many people organise just themselves or on behalf of the department, and sometimes we do it through secretaries as well, so we're quite easy. We've had placements across all different kinds of departments over the years, so basically if you think you might want to take any pupils, any number, just let us know. If it's something, if you're in Ninewells or your surrounding area, even Perth and St Andrews, we're quite interested. We often send people to all different kinds of places. So if you're interested and just want to ask us a bit more, get involved, this is our email, so medicinsight@dundee.ac.uk. If we don't hear from you, you'll probably still see some emails from us just trying to get as many spots as possible. Thank you very much.
Thank you.
There we go.
Okay, thank you. So, very welcome to the highlight of the DCAT year, and I would suggest, Tom, highlight of the Grand Rounds. So this is the annual DCAT lecture. So this is actually the 6th annual DCAT lecture, and I think it's always useful just to remember who we've had lecturing us over the years. So Professor Faruqi was our first lecturer, an endocrinologist with an expert in obesity. We've had Sharon Peacock, microbiologist, Andrew Hattersley, a diabetologist, David Adams, a hepatologist, and Moira White, a respiratory physician. And this year we are delighted to have Professor Sarah Tabrizi presenting to us, who, as you can see, is a neurologist. Sarah was actually Edinburgh born and bred, and this is her first time to Dundee, which is fairly shocking, and is a graduate of Edinburgh Medical School. She escaped down south and has really been at the Institute of Neurology pretty much ever since, where she did her PhD, funded by the MRC, and then a clinician scientist fellowship. You're about to hear that her research focus is on Huntington's disease, and her work is really, truly translational, from molecular mechanisms through to developing novel therapies and clinical trials. I think particularly fitting for this lecture is that Sarah has an ongoing, very active teaching role in undergraduate and postgraduate medicine, and tells us that she is a mentor for 12 trainees as part of her role as a fellow of the Academy of Medical Sciences. So I think without further delay, let me invite Sarah to give the 6th DCAT annual lecture.
Thank you so much for inviting me, and it's great to be back in Scotland. Although I've been through Dundee many times, I haven't actually visited, and it's a beautiful city. So I'm going to tell you about the work that on Huntington's disease that I do, and I'm going to focus it more on the translational research and the work that we're doing to try and develop therapy. So I'm now the director and the founder of the UCL Huntington's Disease Centre, and we recently recruited Jill Bates from King's, who was one of the people who cloned the Huntington gene, and I did part of my PhD with her. And I think the aim of the centre is really to develop therapies, and so we do very much basic bench science, experimental medicine, and then develop and test therapies for trials. And so just briefly, I'm going to talk a little bit about Huntington's disease, and then I'm going to talk to you about the work that we've been doing on identifying genetic modifiers and why this may be important for new therapeutic targets. I'm going to really focus a lot of my talk on the last 5 years of my research, which is leading up to the first clinical trial, which we're doing at the moment, of an RNA-targeted molecular therapy. And then the organisers asked me to give a few slides on my perspective as a clinical academic and how to survive. So I've got a few slides on that at the end. So George Huntington wrote one paper in his life at the age of 21 as a medical student, and he described Huntington's disease. And the reason he wrote it at such a young age is that his father was a GP in Long Island, just outside New York, and he used to go on his father's GP rounds, and there was lots of families with this disorder, which was a disorder characterized by shaking and dementia. And he wrote it, this is a beautiful, it's a bit like James Parkinson's essay, but George Huntington wrote a really beautiful monograph. It was, and he wrote, if the thread is broken, then the grandchildren of the original shakers may rest assured that they are free from the disease. And that was 1872, before Mendelian genetics. And he clearly, very astutely notes the dominant inheritance. So Huntington's disease is the commonest genetic dementia worldwide. It's dominant, the mean age of onset is 40, there's a movement disorder, a dementia, and prominent neuropsychiatric symptoms. The prevalence it's a rare disease, but it's not so rare. It's about 1 in 8,000, and actually the prevalence in Scotland is quite a bit higher than that. And about 8% of new cases have no family history. That's the wrong way. So the mutation is in the first exon of the Huntington gene, and if you inherit more than 40 CAG repeats, you will develop the disease. And it's a large protein that's expressed throughout the whole body. And so there are many different cellular mechanisms of pathogenesis that cause Huntington's disease. And I'm not going to really focus on this, but importantly, and actually it's interesting, there's in protein misfolding diseases that cause Alzheimer's and Parkinson's and ALS and Huntington's disease, the downstream cellular pathogenic pathways are actually quite similar. And in Huntington's disease, there's an important role for protein aggregation, there's dysregulated transcription, there's synaptic dysfunction, mitochondrial toxicity, and axonal transport impairment, familiar in many of the protein misfolding diseases. And the reason I'm showing this slide is it'll become relevant when I come on and talk about therapies. And so this is a poor quality video, but I show it because this is what chorea can look like. And many of our patients who have very severe chorea, it's a movement disorder, have to be nursed on mattresses on the floor, and it's very disabling. So this is an example of ballistic chorea, but chorea comes from the Greek word to dance, and you can see that these are very disabling movements. And actually, again, it's a poor quality video, but I use it because it shows you that the chorea then disappears, and people then become completely rigid. They're unable to speak, unable to swallow, unable to move, and this period lasts for about 5 to 6 years. And this is advanced disease. It's a terrible disease. And in this period, this is very common, that there is often, that you may hear about in court cases, where people want their families to take them to Dignitas and they make advance directives. And there's been quite a few court cases regarding end of life with patients with advanced disease, because it's not a rapidly progressive disease. And then there's also juvenile patients with Huntington's disease. And the youngest person I'm looking after at the moment is a 7-year-old child whose father had Huntington's disease in his 20s. This is a patient who's 16 at the time of this video. You could just see behind me his mum. Her husband died of Huntington's disease in his 30s. Ammar was 16 at the time of this video. He died when he was 21, and Ammar's sister also died of Huntington's disease. So Ammar's mum, who I still keep in touch with, lost her husband and both kids to HD. So it's a devastating neurodegenerative condition, and it's genetic, so it blights families. And so when you do clinics every week seeing patients with Huntington's disease, and I've been doing clinics for over 20 years in HD since my PhD, all of your work wants to focus on developing a therapy, and that's what I'm going to talk about. So I'm going to tell you a little bit about HD genetics because it's interesting genetically. So if you have less than 29 CAG repeats in your Huntington gene, you're completely normal. But between 29 and 35 CAG repeats, it's not pathogenic to the person carrying those repeats, but they can expand into the disease range in future generations. And this is particularly through the paternal line. And that's why all the children with juvenile onset HD most commonly inherit the gene from their dad. And so it's very unstable during spermatogenesis and can expand into the pathogenic range. And that's why 8% of cases don't appear to have a family history. And it's usually because a parent, or one reason, had an intermediate repeat. And this is, if you have more than 39 repeats, you always develop Huntington's disease. It's fully penetrant at 40 or more repeats. And so one of the things that's been of interest is the CAG repeat length modifies the age of onset, but only a percentage of the age of onset. So there's always been a big interest on what are the other modifying factors. And so this is something that I've been interested in, particularly because they're potentially new therapeutic targets. And so this is the age of onset, and this is Huntington's CAG repeat length. And you can see here there's a wide range. The longer the repeat, the younger the age of onset. But if you take 42 repeats here, you can see the wide range of ages of onset. And so one of the things that we were interested in is what modifies that. And so the modifying factors could be environment, and I think that's important, or other genes. And so a big collaborative effort that I was involved in was identification of genetic factors that modify clinical onset of Huntington's disease, and this was published a few years ago. And ongoing work in this area has identified FAN1, which is on chromosome 15, as a key modifier of Huntington's disease. And the mechanism of how that modifies HD is something I'm going to talk a little bit about. So the pathway analysis in the genetic modifier study came up with some very interesting findings. The most important pathways that came up was in mismatch repair and DNA repair, particularly, and that hadn't really been of much interest, particularly in Huntington's disease before this. And so one of the things that we decided to do was there was data on age of onset, but we wanted to try and find out what modified disease progression, because clearly that's important as a therapeutic target. And so we decided to look at deeply phenotyped patient cohorts to identify genetic modifiers. And I think this is relevant to anyone who works in a genetic disease. And actually, the reason we got interested in this is that Andrew Singleton from NIH had done some beautiful work in Parkinson's and looking at what modified LRRK2 Parkinson's patients. And this is some of the work. I had a meeting with Andrew, and this is where some of this came from. So there's limitations in the age of onset for genetic modifier work, because you have to go back to case notes, and the clinician judges when the age of onset would have been from the case notes. So it's got quite wide error rates. And so if you have very accurately deep phenotype cohorts, it improves the power to find association in genetic studies. And that's what we wanted to do to look at disease progression. And so what we did is that I was the PI of a study called Track HD, which was a natural history study which we set up in 2007, which was to try and understand the neurobiology of pre-manifest and early Huntington's disease. And I think really good natural history studies and deep phenotyping of patients is critical if you want to find biomarkers, but also understand the disease better to develop therapies. And I think not all of the reasons, and I think it's a subject of a talk in itself, but a lot of the failed Alzheimer's trials come from previously not understanding aspects of the natural history well enough. And so what we wanted to do was to really try and understand the natural history of Huntington's disease. And this study, TRACK HD, was really to try and understand the neurobiology of the pre-symptomatic phase and early disease, identify what predicted onset and modified progression, and we wanted to establish a clinical trial methodology for this group of patients to do clinical trials that were meaningful. And so we ran it like a clinical trial with independent analysis. There was 4 study sites, and basically we studied 360 subjects, 123 controls, 120 gene carriers who were completely well, pre-symptomatic, and 123 patients with early disease. And we really threw the book at them. We did everything. And they had visits over 4 years, and we published the data in a series of papers. But I'm not going to really talk about TRACK HD, but what we did with the data set is that I wanted to use this data set to see if we could identify what modified disease progression. And so we took the data from the TRACK HD data set, and we very simply put together a number of variables. This was to do with, we did this with our statisticians, and they did a principal component analysis using imaging, cognitive, motor progression variables, and came up with a single progression statistic that was based on a large amount of data, a single unified progression statistic that was a measure of progression. And it was a lot of work, and this is a very brief summary of it, but it was an important measure of progression that could be used for a genetic association study. So when we did that, TRACK HD wasn't so big for a genetic study. We had to validate our findings in a larger cohort, as you have to do with genetics. And so we went to the registry database, which is 10,000 individuals, much looser data set. I was going to say dirtier data set, but it's basically there's not that much clinical information. They had a few cognitive tests, they had motor testing, and were seen once a year, but none of the deep phenotyping that we did in TRACK HD. But the stats team that we work with did a huge amount of multiple imputations of missing data, and they did come up with a progression statistic. I'm summarising a large amount of work, but they were able to use the registry to come up with a progression statistic that was similar. So why was this important? Because we then did a GWAS to try and find what were genetic modifiers of disease progression. And when I was first, my PhD was in biochemistry, so my background is not in genetics. So when I was trying to get funding for this work, it kept getting turned down by everyone because they said, oh, well, the TRACK HD data set's only 250 people. It's way too small to do genetics. So it was interesting because we didn't expect to get a result, but it just tells you how powerful deep phenotype cohorts are. So we did a GWAS on the TRACK HD data set, and we saw a clear signal in chromosome 5. And then we did GWAS in the registry data set, a much larger number, and we saw co-localization with the same variant. And then we did a meta-analysis which strengthened the signal on chromosome 5. And what underpins that signal on chromosome 5 is a mismatch repair gene. And I think I'm going to tell you why I think this is important. So we did a pathway analysis which highlighted again DNA repair and mismatch repair. And so how could this DNA repair and mismatch repair relate to Huntington's disease? Well, one of the things which I mentioned in the genetics is the CAG. And the CAG expands during a lifetime of an individual. And this originally came all from mouse work. And this is just from data from a Huntington's disease mouse model at 10 weeks of age and at 21 weeks of age. And this is the striatum, particularly affected area. And this is the CAG repeat. And as you can see, as the mouse ages, the CAG repeat is becoming unstable and is expanding. And this is called somatic instability of the CAG repeat. And what we now know, and there's increasing data, is this occurs in postmitotic neurons in the brain. And so the relationship to DNA repair is that the mechanism is likely to be relating to somatic CAG instability, because the CAG expansion in postmitotic neurons is possibly modulating disease. And so we've got quite excited about this, and we just recently got a large foundation program from the Dementia Research Institute to study DNA repair and the actual mechanisms of MSH3 and FAN1. I'm not going to talk so much about the FAN1 work, modulating Huntington's disease biology. And so what we think is happening is that MSH3, which is the mismatch repair, is modulating patients via somatic instability of the CAG repeat tract. And what we think is happening in postmitotic neurons is that you get DNA damage, many reasons, oxidative stress, excitotoxicity. The DNA damage is then recognized by this complex MSH2, MSH3, PMS2 and MLH1, which is a mismatch repair complex. And then you get erroneous repair, which results in expansion of the CAG. And longer CAGs are very bad for cells. And what we think is happening, this is progression of Huntington's disease, and what we think is happening over the lifetime of a patient is that there is somatic expansion of the Huntington's CAG repeat tract in the brains of patients. And so this has now become a huge new focus area of work. And you might say to me, well, how does this relate to therapies? And just before I say about how I think it's related to therapies, we also looked at spinocerebellar ataxias and polyglutamine disorders, and I did this together with a colleague of mine in Cardiff, Leslie Jones, and we found that the same DNA repair pathways that were in the GEM paper that I showed you, the Cell paper, also modulated onset in polyglutamine diseases. So that's spinocerebellar ataxia diseases and SBMA and DRPLA. So actually it looks like it's a common underlying mechanism for triplet repeats. And I've just set up a collaboration, well, it's a long-standing collaboration now for a few years, with Darren Monkton in Glasgow, who was one of the team that cloned the myotonic dystrophy gene. And it looks like mismatch repair and MSH3 may also be underlying progression and modulation of phenotype in other triplet repeat diseases that are not CAG repeat diseases. So it's a huge area now of DNA repair and triplet repeat diseases in neurological disorders. And actually triplet repeats cause a lot of neurological diseases. So it's become a big area of interest. So just conclusions for this part of my talk are, I think you really use deeply phenotyped cohorts. We got a signal in 216 patients that was significant. And as I say, Andrew Singleton has done the same with LRRK2 cohorts in Parkinson's. So if you have very deeply phenotyped cohorts, the GEM-GWAS paper in Cell was 6,000 people, because age of onsets are very, very, it's not such a precise measure. So MSH3 is likely to be a genetic modifier of Huntington's disease, and that DNA repair pathways modulate disease progression. And this is in a monogenic disorder, showing the importance of additional genetic modifiers. And so we found association in just 216 subjects, which was replicated and strengthened in a meta-analysis, which suggests that progression is a good measure, that our variant has a large effect size, and it really supports the use of very deeply phenotyped cohorts of patients. And that's why doing studies like that in patients is of enormous value in understanding disease. And actually a lot of our work has come from observations in patients and then moving back to the lab to verify them. And so we've also shown that DNA repair pathways modulate age of onset in polyglutamine disorders, which I think is likely to involve somatic instability. And interestingly, MSH3 is not constrained by selection. And that was in the recent Nature paper, the EXACT paper. And so that's good, because what we want to do is develop MSH3 inhibitors. And we know from previous work that's been done with MSH3 that if you knock out MSH3 in mice, you abrogate somatic instability. And so part of the DRI program that we're developing is to understand the biology of this better and to develop potential MSH3 inhibitors, because if it's not constrained by selection, inhibiting it may not have adverse side effects. And by lowering MSH3, We hope to prevent somatic expansion of the CAG during the lifetime, and it may be a good target for a potential treatment and also for other triplet repeat diseases because it may stabilise the CAG because erroneous repair results in expansion. So this has become a really big area of research now in my lab, and all from the original genetic studies. So I just wanted to use that to illustrate how going from patients to genetics, then back to the lab, can give insights into developing therapies. So with Huntington's disease, many patients with Huntington's disease about 20% of those at risk decide to have a predictive test. And a predictive test tells you whether you're going to develop the disease in the future. Only about 20% of people go for a predictive test because we don't have a therapy. And it's a very personal choice to find out that you're going to develop a pretty awful neurodegenerative disease in the future. And these people have often looked after a parent with the disease, and they know they're at risk. So and actually I teach on a master's course in ethics, and one of the course talks is it ethical to do predictive testing in people when you don't have a therapy? But importantly, people who have the predictive test, who are well, many of them come forward for our research, and many of them were in the TRACK-HD study. And I think what's important is that this allows us to understand the preclinical phase of neurodegeneration. We know from a lot of our work, which I don't have time to show you, that the disease begins 20 years before people show any signs of the disease. The disease has already started in the brains of patients. And so this is probably very similar for Alzheimer's and Parkinson's, and that the disease takes a long time to express itself. And so what we want to do in Huntington's disease is really use the disease to exploit the genetic certainty to understand the preclinical phase of neurodegeneration. And it's a model disease in that respect, because if we can nail a treatment for Huntington's disease and show that we can reverse brain disease or modulate brain disease, and I think that gives great hope for other neurodegenerative diseases like Alzheimer's and Parkinson's in particular, because it shows you that these brain diseases are tractable because there are no current therapies that modulate the disease. So HD can be thought of as a model genetic disease in that way. So, a person is born completely well, and then at some point they start developing signs of the disease. And they're born essentially with healthy neurons that become dysfunctional and then die. And so if we can really understand this phase of the disease and develop therapies, because we have a genetic test, we can potentially treat people 10, 20 or 30 years before they show signs of the disease and prevent the disease ever occurring. So I'm going to come on and talk about the program of therapy that I've been working on, and this I think is important, and that's why I showed you the downstream pathogenic pathways. I am a great believer in treating proximally in these diseases. I don't think if we treat some downstream cellular pathway, you're going to have a big impact on the disease. I think there's too many downstream cellular pathways. I think it's like trying to plug a bath with multiple plugs and water is pouring through. You just want to plug right at the beginning, because that's really where the disease begins. Because these are all the current priority preclinical therapeutic targets for HD, and you can see there's loads. But I think really on my money, we need to target the DNA or RNA, because then it doesn't matter what all these pathways are, you target at the beginning. And I think that's where a lot of the focus of my work is, all on Huntington lowering. So I'm going to just talk you through the current Huntington lowering approaches. So this is Huntington DNA. It's transcribed, the Huntington pre-mRNA, the splicing. This is the cytosol. It's Huntington mRNA translated to the protein, and then there's an exon 1 protein as well, which is highly toxic, which Jill Bates also described. So I'm going to tell you about antisense oligonucleotides, which we've been working on, which bind to Huntington pre-mRNA. There's RNAi, which many of you will have heard about, which targets mRNA. Small molecules, challenging in development for trying to target Huntington splicing. Zinc finger proteins are in development to target Huntington DNA, again very much in preclinical development. And CRISPR, which everyone talks about, can target and cut out the CAG, but many of these other approaches have huge issues to do with delivery. And CRISPR to treat the whole human brain is light years away, and I'm happy to discuss why. So there's a lot of challenges to treating neurodegenerative diseases, because it's with what, when do you treat, where do you treat, how do you treat. I've just given a number of the approaches. And I think importantly, and I think this was really important for me, because I would have had lots more sleepless nights than I did, is it reversible and can the dose be titrated? And I think that's important. So the big issue with treating brain diseases is the size of the human brain, which is very large. And so there are hundreds and hundreds of papers treating mice whose brain is this big, which is the size of your pinky nail. It's quite easy to treat a mouse brain with a viral vector. You can actually cure a mouse of Huntington's disease, but then you have to translate it to an organ this size. And these neurodegenerative diseases target the whole brain, so you can't treat one small part. And so this is a small monkey brain, this is a large monkey brain, and this is the human brain. And if you really want to translate, you have to do work in non-human primates, and then you have to do work in larger animals. And targeting the human brain is the big challenge in neurodegenerative diseases. So in Huntington lowering, the kind of therapeutic delivery strategies are a number of strategies. All of them are in preclinical development, but I'm going to tell you about one that is in a clinical trial at the moment. So the single agents, which are transient, like antisense oligonucleotides or siRNAs. There's gene therapy. This is still in preclinical development for Huntington's. And the thing about gene therapy is it's a permanent expression of an agent. It is integration of genetic material. It's a permanent expression, and it's not reversible. So you've got to be very, very sure before you inject it into a human brain. And this uses mostly AAV. Their zinc finger program is moving forward, and I'm very involved in that, but it's still very much in the preclinical program. And delivery methods to the CNS are currently intrathecal, which you inject it into the spinal fluid, and intraparenchymal, which are injection directly into the brain. The AAVs, which are being developed, which are injected directly into the brain, are being done in larger animals now. They're being done in large pigs and sheep, but they can still only target a very small part of the brain. So they target, at the moment, it's just targeting the putamen, which is a relatively small part of the brain. It's very difficult to get AAV beyond a small region. So antisense therapy in HD is what I'm going to spend the rest of my talk talking about in the trial. So this achieves Huntington lowering, but importantly, it doesn't eliminate Huntington. You've always got Huntington present. And it binds, the ASO binds to the Huntington pre-mRNA, and it leads to its degradation. And I think importantly, antisense oligonucleotides are very diffusible. You can titrate the dose. They're stable and reversible. And I'll come on and say why I think that's important. And so I've been working in a collaboration with this amazing biotech company, which has become more famous now because of the spinal muscular atrophy trial, and I'll show you some of that data, called Ionis Pharmaceuticals. And so they have developed over the last 20 years amazing technology of antisense oligonucleotides. And their chemistry is very interesting and important, and they've done a lot of work to refine the chemistry. So this is the DNA. The chemistry has an MOE moiety at either side and allows affinity, stability, and tolerability. And then there's a 20 base pair oligonucleotide, which is recognized, and then the DNA RNA is degraded by the RNase H1 substrate. And the MOE moieties allow the stability of the antisense. And actually, this is essentially like a drug. And so the antisense was years of screening for Huntington antisense. But basically, it's a single I just like this video, that's why I'm showing it. It's a single strand of chemically altered DNA that binds to the Huntington pre-mRNA, and then through Watson and Crick binding, it's degraded. So intrathecal ASO drug delivery to the CNS, they don't cross the blood-brain barrier because they're large, and so you have to give them intrathecally. And I think there's a precedent for intrathecal drug delivery with anaesthetics and chemotherapy, and importantly, they have long half-lives. And so the drug is delivered into the spinal fluid, which is then, it diffuses very rapidly to the brains to the cortex, and actually that is the work that we've been doing in the Huntington's program, and I'll tell you about that. So I'm just going to quickly tell you about spinal muscular atrophy, and this is why this is important. So spinal muscular atrophy is a terrible disease of children. Type 1 is the infantile, very severe. 80% of the cases of spinal muscular atrophy, they get the disease at less than 6 months of age. They have very short life expectancy. Most don't live beyond 2 years of age, and they're never able to sit or reach motor milestones. And the severity of the disease is related to the copies you have of a sister gene that takes on some of the function of SMN1, which is lost. And this is SMN2. So type 1, the most severe form, have the lowest levels of SMN2. So what Ionis have done, and this is a lot of data, they developed an SMN2 targeted antisense for SMA. And basically what the ASO does is upregulate the sister gene SMN2 to take on some of the function of the missing gene SMN1. And Adrian Craner at Cold Spring Harbor discovered the SMN2 gene and he's an RNA biologist, did some beautiful work. So 250 children were treated. Some were on the drug for up to 4 years. There was no safety or tolerability concerns. They demonstrated target engagement, and they did a lot of work in non-human primates. And so this was a huge press release at the time, and it was big news. For the first time in a neurological disorder trial, the trial was halted early at an interim time point because it was unethical to continue with the placebo group because the drug was so effective. And so the trial was halted early, and the FDA gave its fastest approval ever for any drug within 3 months for rapid review. And I'm going to show you a little bit of the data because it's remarkable. And the data actually was published last week in the New England Journal of Medicine from the phase 3 ENDEAR study, with the one that was halted early. So they did the first trial. This is motor development score, motor milestone score of children, and higher is better. This is visit days, and this is infantile spinal muscular atrophy. And so the very first trial was in red, which was an open-label phase 1-2, and they used historical control children. And it was open-label because they didn't want to, at that time, to do a placebo. And so it was very clear that the drug was having a beneficial effect, and this is 2 years, and the children were achieving motor milestones. This is the study that was halted early. These are the children that were getting the intrathecal ASO, who were achieving motor milestones, and this was the group of children that got the sham. And you can see here, by this stage, they were had either died or were unable to continue because they were very, very, very sick. And this was when the trial was halted because there was such a huge difference. So the drug allows these children to achieve motor milestones. They're able to sit. I've seen a video of a little child with infantile SMA riding a bicycle. But this is where I think is the most important data, and this is not published. So Ionis and Biogen decided to, if you have had a child with SMA, it means that both parents carry the SMN1 mutation. So you can screen children at birth, subsequent children at birth. So this was done. If you had a child that had SMA, then you could offer screening at birth to the parents for the subsequent kids. So the children were screened at birth or in utero, and then they were offered, at 1 or 2 days old, the opportunity to go and have intrathecal ASO drug with nusinersen. And this is what happened in the NURTURE study. The disease never manifests itself. So this is the NURTURE study. It's a pre-symptomatic infantile onset SMA. And this is normal motor milestones. So these children who are usually dead by the age of 2 years of age and at the age of a year are on a ventilator achieve normal motor milestones, and you've completely prevented the disease. It's remarkable. And so this has been of huge excitement in neurological diseases because this is showing us that what these kind of RNA-targeted therapies can have remarkable effects. And so the major questions we had for our ASO, and I'll go through this fairly quickly for time, was that we wanted to design an ASO that reduced Huntington mRNA and protein, one that was safe in preclinical toxicology. We wanted to do a non-allele specific approach for a number of reasons, because we wanted to be able to treat everyone. And could we intrathecally deliver ASO to the brain regions affected by Huntington's disease? And this is just data showing that antisense oligonucleotides infuse into the CNS. They get taken up by neurons and astrocytes. Equally, they're highly diffusible molecules. And one of the experiments that we did, because early on we started discussing this in 2010, was that we needed to dose an animal with a very long spinal cord. And so there's these pigs called Yucatan pigs that have a spinal cord of over 50 centimeters. It's very long. And so the pigs were given an intrathecal dose of the ASO, and then they were sacrificed, and we measured mRNA levels after the ASO delivery. And this is Huntington mRNA. And you can see here, with an intrathecal delivery, lumbar intrathecal in pigs, you can see here in the occipital cortex, the temporal cortex, and the frontal cortex, there's very good Huntington lowering. So the ASO is diffusing upwards. So if you inject into the lumbar intrathecal region, CSF flow is upwards. And so I mentioned about non-human primate work. I think for developing diseases for brain disorders, you have to do non-human primate work. The CNS of a non-human primate closely resembles our CNS structurally. And so what we did here was that we dosed non-human primates 4 times at monthly intervals, which is the current regime as this current trial. And they had lumbar intrathecal dose, 1 month, 2 months, 3 months, 4 months, and then the animals were sacrificed. And this was 1 week post the last of the 4 doses. This is RNA and protein. And you can see here very good Huntington lowering, over 80% in the frontal cortex and occipital cortex, key regions affected in Huntington's disease. About 50% lowering in the chordates, very deep brain structure. In the thalamus, again, a deep brain structure, about 50% lowering. And hippocampus, about 75% lowering. And what this data shows is how long the drug effect lasts, so how long the effect on your target lasts, the pharmacodynamic effect. And this was 8 weeks after the last of the 4 doses. And you can see here there's a sustained pharmacodynamic effect on Huntington. So the drug effect lasts for a reasonable length of time. Again, in the caudate, it's maximal at 1 week, but there's still about 15% lowering at 8 weeks. And so what we really have is a cortical predominant pharmacodynamic effect on Huntington, and it lasts about 8 weeks. And so this was the data we needed to be able to go to a human trial. So we identified ASOs targeting Huntington that were safe in mice. They improved phenotype and survival in HD mouse models, and I don't have time to show you all the data. We found that long-term suppression of Huntington in the non-human primate CNS was well tolerated, and we had widespread distribution. And we did a lot of toxicology studies in a large range of large animals, and we allowed a sufficient safety margin for us to go to doses in the clinic. And so the clinical development plan was, would the ASO be safe and well tolerated in patients? Could we get evidence of target engagement? And what's the best study design? And we spent a few years designing the study. Unlike the SMA study, we decided to have a placebo group, which we had a lot of pushback for a first-in-man study. But just over 2 years ago, we dosed the first patient with the very first ASO targeting human Huntington mRNA in September 2015 and in London and in Vancouver, which is one of our study sites. So it was a big event for HD. And so the objective of our study is to evaluate safety and tolerability of increasing doses. We want to know how long the drug stays around in CSF, the pharmacokinetics, because that's important for future trials, because we really don't want to try and have to dose monthly forever. And we also wanted to look and see if we could measure a biomarker of target engagement, which was a level of mutant Huntington in the CSF. And so we spent a long time deciding to design the study. It was a phase 1 first in man, but we linked it to a multiple ascending dose as a 2A study. And the MHRA and BfArM in Germany and Canada Health all were very, very open to having this slightly unusual design for a first in man study. We have a placebo group, which we felt was important. Randomized 3-to-1 active to placebo. We give the drug by an intrathecal bolus, lumbar intrathecal bolus, and the patients get 4 doses, like the non-human primate study, and then they're followed up for 15 weeks. And Roche have partnered the program, because actually another thing I've learned about trying to be involved in developing therapies, if you really want to develop a therapy that you want to take to hundreds of people worldwide and do a phase 3 study that's going to cost $150 million, you have to get a big pharmaceutical company to partner you. Roche are partnered, and we hope they will opt in and do a worldwide phase 3. So this is the design. We had 5 cohorts, and we had an independent data safety monitoring board who would look at the data unblinded at the end of each cohort and tell us whether we could go to the next cohort. And I'm pleased to say we've completed enrolment and finished enrolment of cohort E. And this was our target engagement biomarker. We had to wait to start the study till we had this, and we published this a few years ago. And this is a measure of mutant Huntington in the CSF, which we measure using this very, very sensitive single molecule counting assay. But this is what we're using as our marker for target engagement in the CSF. And so, unfortunately, I haven't got the data to show you yet because we're wrapping up, but the first patient entered in June, enrolment was complete, and we opened an open label extension. So all of the subjects who were in the original trial are going to get the active drug, and I think that's critical if you've been in a placebo group. And so everyone will get the active drug, and we're about to start an open label extension, which is about to start dosing in the next week or 2. So in the end, we dosed 46 patients with early stage patients, so they could give full informed consent. We did in UK, Germany and Canada. The health authorities were very supportive in these countries. Recruitment wasn't challenging. There is a huge unmet need in Huntington's disease, and we had motivated physicians and study teams, and we've completed the dosing phase of the study. So the trial's been executed with high standards. We enrolled the intended early stage patients. The intrathecal dosing was very well tolerated. We've had no safety concerns to date, and the profile supports further development. And we're just following up the last number of patients, and I'm going to present the results in the new year, and writing it up for publication. I'm actually starting to draft the manuscript. Enrolling patients from the first study in the open label extension, and this allows us to get long-term safety data, and we hope to announce the results of this study in January time, and if the first study meets all those objectives, then we're hoping that Roche will opt in to take it forward for a worldwide several hundred patient phase 3 study, because we want to go straight to a pivotal trial rather than do an intermediate, because we don't need to do a dose finding because we did the multiple ascending dose design. And that's the hope, that we will go forward to a worldwide phase 3. And I think ASOs are quite exciting time now, because if the Huntington's trial is positive, then it'll tell us you can get these drugs very effectively to the adult human brain through intrathecal dosing. I've told you about the SMN trial. It's got the unfortunate names of Spinraza, which Biogen named, and that's the SMA drug, which has been revolutionary. As I say, that came out in New England Journal last week. There is a SOD1 ASO in development for ALS. There's one in clinical development against tau, for tauopathies. There's preclinical in development for alpha-synuclein for Parkinson's. Ataxin 2 is in preclinical development for SCAR2 and as a modifier for ALS, and there's in preclinical development against the prion protein C9orf, Ataxin 1, Ataxin 3. So I think it's like John Hardy, who has the office next door to me. I think he says the HD trial is positive. I think then ASOs for neurological and neurodegenerative diseases for adults may open up a whole area of therapeutics. So this is where this is, and I will hopefully be able to share the trial results in the new year. So this is my final slide. I think for someone who works on Huntington's disease, and it's a really desperately sad disease of families, I couldn't keep doing clinics if I wasn't developing a therapy that I hope one day that we would end up developing a therapy to prevent it. And so I have a big was a strategic award, but now a collaborative award from the Wellcome Trust to develop therapies eventually to treat 18-year-olds who are 20 or 30 years before their predicted onset. Because I think if we could intervene then, when people are completely well, we may be able to change the course of this and prevent the disease completely. And that's the goal. And then I could retire. So that's the plan. So my career path, I was told to give a bit about my career path, which Ewan summarised. I did biochemistry, then medicine, and then I did a PhD, 10 years as a clinician scientist, and set up my own group. And my advice for survival as a clinical academic. So I think you have to learn coping strategies to become resilient. And I think that, for me, and all the people I mentor, is the most important tip for survival. Because I think that when I was a clinician scientist, and I had sent this paper off to EMBO, and I got a really vile review of my paper that I thought was really important. And I was very, very upset. And I went to talk to Charles Wiseman, who was my clinician scientist supervisor, who's a very famous scientist. And he said, you have to become more resilient, and you cannot cry when you get a very horrible review of your the paper that you think is very important. So I think that, to me, is the most important thing, because however good you are, everyone gets paper rejections, and however many grants you have, you will always get a grant rejection. And you also will have difficult colleagues. So I think my most important tip for survival is to become resilient. And that's, I think, the most important thing I've learned. And I think you need to have a lot of focus and tenacity and never give up. I was nominated as a UCL academic role model, and they had to say, what do you think is really good advice? And I actually mean it. It's the Winston Churchill never, never, never give up. And I think that's right. You absolutely have to believe in what you do. I think you have to believe in what you do, and you have to be passionate about it. And mine is finding a cure for Huntington's disease. And I also think, and I think this is important, you've got to try and be as nice as you can be as well, because I think it's quite difficult trying to be a successful clinical academic in a very environment where you're trying to do everything that's very competitive. And so I think it's important to try and be nice, but don't let yourself be kicked around. And I do think you need to make your work as fun as possible, and you need to enjoy it. And the other ones, I think I still love doing clinics. They're good for grounding and motivation. I travel a lot for work now, so quite a lot of them end up being, I'm not there a lot, but I do think it's really important, because you go and do a clinic, and however bad, things don't always go well in the lab or on research, and you go and do a clinic and it reminds you why you do what you do. So I will never stop doing clinics. I do think you should ask for advice and mentoring as you need it. I think that's important. I think getting a good mentor. And I'm very keen on inspiring and mentoring and training the next generation. And I think that's really important. Even like one of my PhD students is now PI in the HD Centre, and I said to him, you've got to always be thinking about future-proofing, because you never know what will happen, and you have to future-proof. But also, if you have lots of young people around you who are smart and clever, they do keep you thinking and motivated. And I really enjoy my lab meetings, because I've got lots of really clever PhD students. And I think this is really very important. And I think as you get older, and I really feel this as I've got older, your work is important, but you mustn't forget your loved ones. I think that's probably one of the other most important things. They really are the most important people in your life, and not your work colleagues. So thanks to you for listening. Thanks to the patients and families who give all their time and dedication to our Huntington's disease studies. This is thanks to all my funders. And this is an old picture now. It's just my side of the group, and this is my research group, and lots of women. So I'm quite into girl power. Thank you.
So thank you very much, Sarah. That was a truly inspiring talk, and I think we will all be waiting avidly for those results in January, and all our fingers are crossed. So we have time for maybe 2 or 3 short questions, and Tom is going to run around with a microphone if anybody wants to. And I will start, and hopefully this doesn't portray my poor understanding of biology, but is there a potential for the size of the triplet repeat to affect the efficacy of the antisense drug, or will it? And have you looked into that at all?
It's a very good question. And actually, we didn't design the ASO around the CAG repeat for that reason. So people are trying to develop ASOs targeting the CAG, and the zinc finger is targeting the CAG, but the problem about that is that there's lots of CAG and transcription factors, and you end up with terrible off-target effects because they bind to other important transcription factors. So one of the reasons for the very long screening is that we're actually got an ASO that binds exon 36, which is halfway along, and so it's quite far away from the CAG for exactly that reason. So
Okay.
it's a very good question.
Good. Thank you. Yes. Hi.
Hi there. I've got 2 questions, if I may.
Yeah.
Firstly, to echo what you were saying about the nusinersen issue. I look after a 15-month-old infant who had a diagnosis made at 8 weeks, and I never, ever thought I would see this in my professional lifetime. It is absolutely astounding.
Very moving, actually. I went to a meeting
Yes.
recently with Richard Finkel. Adrian Craner presented the SMN2 RNA story, and then Richard Finkel presented all the videos of these kids in the trial, and it was really moving.
And it's awful to have to speak to these parents, and you're telling them they're going to die, and they die.
They do.
All die. And it's quite astonishing. So my questions were, firstly, around what your gut feeling is about the length of time that you would have to continue this treatment, accepting that it's intrathecal.
Yeah.
And of course, that's a question that these parents have, and clearly applies to your patient group too, if it's successful. And the second is I suppose, a wider question. I got this drug for nothing on the expanded access program, and then it was licensed by the FDA. And the next day, in the press, came out the cost, and the cost for the first year of treatment was $750,000. And we cannot possibly afford that in the National Health Service. I was racked with guilt. Well, it hadn't been paid for, but when the next child comes along and the next adult, if this is successful, we're going to be faced with very similar costs. And I'd just be interested on your views on all that.
Yeah, they're 2 great questions. And actually, the Lancet Neurology did an editorial recently on the cost of this drug, on the cost of these drugs. So nusinersen is currently $750,000 in the US for the first year of treatment, and then $150,000 for every dose after that. So huge. And I think it's really important issue. It's not going to be affordable in this country. And the HD, we've already started discussing this for Huntington's disease, for therapies that are these sorts of molecular therapies, and how we're going to pay for them. And I think it's a hugely important area, and they're going to have to be a lot cheaper for us to be able to deliver them. At least, I talked to Mike Rollins, who was the chairman of NICE, who's a friend of mine, and he said, there's got to be cheaper than £100,000 a year, much cheaper than that. And so that's our big challenge, is so we can develop these therapies, we've got to pay for them. And then I think they are lifetime therapies. So I think with the nusinersen and the HD trial will be probably similar after we get the pharmacokinetics, the pharmacodynamic data, but that you get 3 months of loading, 3 to 4 months of loading, monthly loading, and then the drug is given every 3 months forever. And HD is likely to be similar if this moves forward. So it's a drug that's given forever, and the cost of that, and delivering an intrathecal drug that has a number of challenges because it's got to be in an aseptic environment. And so I think the two things you've brought up about how long the drug will have to be given is very important, and it is a lifelong treatment, and also cost. How we're going to pay for these drugs. We can develop therapies for these diseases, but we have to think of ways of paying for them. And I don't have an answer to that, and I think it's going to become a very big topic of conversation because of nusinersen is really very expensive, and Biogen's been quite aggressive in its costing.
Okay, I think for interest of time we should wrap up. So once again, if we could just thank Sarah for a really inspirational talk.
Thank you. Yeah, it's true. They end up very expensive. Yeah, it's a really good question.
Well, it's so dramatic to essentially restore...
Actually, these